Well, good night, everyone, and I hope you all are quite well. Well, today actually I came to discuss about cell division, especially mitosis cell division, because at first what is cell division? I mean the division of cell into two daughter cells is called cell division, and you know very well that cell division is cell division. Now I want to talk about mitosis cell division. So let us talk or let's begin with the mitosis cell division. What is mitosis? The process by which the nucleus and the chromosome of a cell or of eukaryotic organism. And here, the nucleus of the mother cell divides only once and make two daughter cells. Well, and maybe the question already that arises that where does it occur? It occurs in the somatic cell of all eukaryotic organism. So, and you know very well about plant cell and animal cell. So it may be in plant cell, it may be in animal cell. So today I have taken the diagram. I have taken plant cell already. The five uh, frame I have drawn. So let's talk about at first that mitosis cell division occurs into two phases. At first, it divides the division of nucleus occurs and then the division of cytoplasm. So there are two phases to for mitosis cell division. So the division of nucleus is called karyokinesis, and the division of cytoplasm known as cytokinesis. So let's talk about karyokinesis. Listen, I can write that. Okay, so the karyokinesis, the division of nucleus, starts with five stages. That's are these are number one, that prophase. Number one, prophase. Then number two, Prometaphase. Number three, Metaphase. Number four, Anaphase and Telophase. These are the stages of, karyokinesis. So in these five stages, the mitosis cell division, especially karyokinesis, just occurs. So the plant cell, and the nucleus. This is cell wall and inner membrane, cell membrane, or actually the jelly fluid, that is cytoplasm, though now I am showing the nucleus, the division of nucleus. So this is nucleus. And you know very well. So if I actually draw, or if I want to show only the structure of a nucleus of the cell, then you just know this is the nuclear membrane, like nuclear. This is actually, I think some of the writings is unclear. So if you don't understand, just write me through comments, then I can read, or I can what is justify that are you understanding, or the written or the diagram or drawing is visible or clear or not. Okay, so listen, this is nuclear membrane, and this is nuclear pole. Nuclear pole. Like this is nucleus. Nucleus. Then these are chromatin fiber. These are chromatin fiber. These are chromatin fiber. And the jelly-like fluid, jelly types of fluid, like in the cell, or outside the nucleus, there are cytoplasm. Or if we think, or if we talk about only nucleus, then inside the nucleus there are jelly-like fluid like cytoplasm. These are called actually nucleoplasm. Nucleoplasm. So actually this is the structure. Actually, whenever just many years ago, the scientist or the biologist, they were researching, uh, they did research about cell. On that time, it was found only the cell membrane, or in the middle of the animal cell, only one nucleus and cytoplasm. But whenever day by day, with higher technology, the electronic microscope has been invented. On that time, the biologist or the scientist were able to see the ultra structure of dead organelles. Okay, so usually know the parts of the cell is called organelles. These are very tiny that we cannot see with our naked eyes. So if we think about only the structure of a nucleus, then you will be able to see there is a dark, dense area in the nucleus is called nucleus. The outer membrane of the nucleus is called nuclear membrane, the nuclear chromatin fiber, and nucleoplasm. So this is actually mother cell that we have taken. Understand? I am just rubbing because the board is not so enough strong, so I have to rub some of the things because I need much more space to make you understand actually. So this is the mother cell, and this is nucleus. Cytoplasm, cell wall, cell membrane, and nucleus. So this is the first stage. Like I'm writing the figure. This is prophase. This is prophase. This is the longest and the first stage of mitosis cell division, especially of karyokinesis. Here, the nucleus becomes bigger in size. So, as my space of my whiteboard is short, that's why I cannot write the points of this description. I'm just telling you, you just try to understand. Okay. So, and if you don't understand, prophase, nucleus becomes bigger in size. And we know, already I have shown that here, in the nucleus, there are some, what is chromatin fiber. So, second points of this stage is that due to continuous dehydration, the chromatin fiber condenses and just uh, it just seam and make thread-like structure called chromosome. And then the chromosome divides longitudinally and into two sister chromatid, and the two sister chromatid join at their centromere. Like this is nucleus and this is nucleolus, and these are the chromosomes. I mean chromatin fiber, and then just oh, one thing I just can't but tell. You have to remember, though just I'm going to show here the five stages. That's why I did not draw here. In prophase, there are early prophase and there are late prophase. There are late prophase. Then in early prophase, that this chromatin fiber, just due to continuous dehydration, the chromatin fiber visibles and just they in late prophase. This is actually like this way in late prophase, nucleolus, and these are longitudinally divides and joined at their centromere. I'm just not drawing here, just I'm just telling now. This is prophase. And prometaphase? What about prometaphase? So in prometaphase, this is actually very short stage. Here, what happened? The nuclear membrane. Here I'm showing the nuclear membrane. Nuclear membrane just disappear. So I'm not doing it clearly because it's just going to disappear, and the nucleolus also disappeared here. And what happened? The due to accumulation of protein fiber of the cell, there is spindle apparatus created. The spindle apparatus created due to accumulation of protein fiber like this way. So are you understanding? I'm just not writing every leveling, like just cell wall, cytoplasm, cell membrane, nucleus. So again, I don't need to write here because this is the basic of the cell. So I'm just not going to that part actually. Well, so here the nuclear membrane and the nucleolus almost disappear, and spindle apparatus already made due to the accumulation of protein fiber of the cell, and the chromatid, sister chromatid, attached with these spindle fiber. I don't know is it clear or not actually. So by this way, these are the spindle apparatus. Now let's go to the third stage, that means metaphase. I can write. So already now I can write because the first stage, second and third, fourth and fifth, prophase, prometaphase, metaphase, anaphase, and telophase. Well, so what happened in metaphase? Let us write figure metaphase. In metaphase, there's no any nuclear membrane, no nucleolus, no nothing. Just the spindle fiber directly and the chromosome, the sister chromatid. Oh, one thing I just goes from one pole to the another pole, equator. Now, all the chromosomes locate comes at the equator, all the chromosomes, and become short and clear and thick, like this way. My duster now is clear. These are the sister chromatic, and here it's very clear. So in metaphase, the chromosome come at the equator of the cell. So let us just review what happened in prophase. It is the first and the longest stage of mitosis cell division. The nucleus becomes bigger and the chromatin fiber due to the continuous dehydration can be visible, and what happened then they divide longitudinally and make two sister chromatid, and sister chromatid attached or joined at their centromere. And then in prometaphase, the nuclear membrane and the nucleolus totally disappear almost, and the spindle apparatus or spindle fiber already created due to the accumulation of protein fiber of the cell. And then in metaphase, the all chromosomes or sister chromatid comes at the equator. Now, what happened? Anaphase. Just going to write figure anaphase. Here, I can tell that again I am drawing the spindle apparatus. This is spindle apparatus. Now that already I told there are sister chromatid. The condition is actually like that. This is actually centromere. So if the cell, it will be divided. So at this stage, the what happened? Sister chromatid divides, split up, and just go to the two respective poles. There are two respective poles. Like that. I'm not so good in drawing, just to make you understand. I'm just trying to do actually like that. Here, what happened? The chromosomes, the sister chromatid divides, split up from their centromere, and here the chromosome reaches to their respective poles. Now just see, they are not at their equator. Now what happened at the last stage? In last stage, that is telophase. Telophase, this is the last stage of, mitosis cell division. This is actually now whenever they are just trying to reach or already reached their respective poles. Again, there are two nuclear membrane, nucleolus reappears. There reappears. And spindle fiber disappear, nuclear membrane and the nucleolus reappears. Already produced. So see, this is the mother cell of a plant cell, and now in telophase, again same types of two daughter cell already produced. This is actually the summary of mitosis cell division. That again, we can go to the review. Mitosis cell division is a division of somatic cells of all eukaryotic organisms. And there are 5 stages. And before that, I can tell that mitosis cell division occurs in two phases. Number one is karyokinesis, the division of nucleus, and then number two is cytokinesis, the division of cytoplasm. So today, actually, I have discussed. I do not know how much I could make you understand or not. So later on, I will try more. So if you want to know, or if you have any query, or if you have any question about that, or if you want to know more details or some of the basic things you do not know, like I'm talking about centromere, I'm talking about sister chromatid, I'm talking about the protein fiber of the cell. So some of them maybe do not know the basic things, so no problem. So if I get the request, so I will try to make you understand later on in my next live or in my next video, no problem. So just see. So there are in karyokinesis, there are five stages. Number one, prophase. Number two, prometaphase. Number three, metaphase, anaphase, and telophase. So in prophase, the nucleus becomes bigger in size, and that due to dehydration, the chromatin fiber uh, condenses and make chromosome, and then they divide and make two sister chromatid and join at their centromere. And in prometaphase, the nuclear membrane and the nucleolus totally disappear. And another, this is called actually spindle. This is called actually spindle apparatus. I can write here, no problem. Spindle fiber or spindle apparatus. So here, and there are two respective poles, and the middle area is called equator. So in metaphase, all the chromosome comes at their equator in the middle of the cell. I mean, not they are spreading all the in all the space of the spindle fiber. And then in anaphase, the chromosome divides, split up, and try to go in their respective poles. And in telophase, again, nuclear membrane, I mean by this way, nuclear membrane nucleolus reappears and the spindle fiber disappears. This is the summary of the karyokinesis cell division of mitosis cell division. So today, I'm just, I wanna finish, so no problem. And today I want to finish here. So all the best and stay happy. Again, good night everyone and have a nice dream.